Good morning or good afternoon, depending from where are you joining us. I would like to thank and welcome you for participating in this webinar organized by ILC Dover. ILC Dover is a worldwide leader in the design and manufacturing of engineered flexible protective solutions to critical applications from aerospace to pharmaceutical industries. My name is Paulo, and I have the honor to host this session about high containment single-use FIBC for pharmaceutical powder handling. The speaker today will be Scott Patterson, Vice President of Pharma and Biopharma Technical Support. Scott has been with ILC Dover over 14 years and leading innovative advancements to the pharma and biopharma industries using single-use containment technology. Today, we will talk about the DoverPac, a reliable high-containment single-use FIBC, which has been invented more than 20 years ago. At this time, the ILC Dover focus was developing and manufacturing the spacesuit for NASA, the core business since 1947. Basically, the spacesuit is a containment system for protecting the astronaut for a very aggressive environment. And based on this experience, going back to 1997, the pharmaceutical Eli Lilly challenged ILC Dover to go behind boundaries and develop the single-use containment system to provide operator exposure levels of less than 1 microgram per cubic meter. The product contact material should meet all international standards, and it should be anti-static and robust enough to be in contact with a wide range of solvents being used in the industry. But before starting, I would like to welcome you all and inform that if you may need to ask some questions, please type it on the question box and click submit. At the end of the presentation, Scott and I will try to address all the questions. So now, welcome Scott, and please talk to us about the DoverPac.
Thank you, Paolo, and welcome everyone. Welcome to our virtual trade show experience as a webinar. We appreciate the attendance this morning. Our overview of the program today will focus on the DoverPac high containment system, which is our FIBC product, flexible intermediate bulk container. We'll give you a little bit of background about our quality by design that has been built in over the years for the DoverPac, and then specifically look at the applications where the DoverPac is used in API processing as well as HPAPI processing through chemical synthesis and oral solid dosage. With that, we're always obligated to show the performance following the industry standard ISPE SMEPAC protocol. So we have some case studies and some data to share. And then along with the comparison of the DoverPac to stainless steel equipment and really looking at cost and performance of using a single-use product over a rigid stainless steel product. As Paolo said, at the end, we'd like to have your questions and any discussion points. So please, as we go, if you have a question, type it in the box and we'll prepare to answer those at the end of the webinar. So again, the DoverPac is a high containment FIBC. Paolo mentioned that the original goal in 1997 was to create a high containment system with containment levels of less than 1 microgram per cubic meter. Currently, the data shows us that the typical DoverPac in a pharmaceutical process will achieve less than 0.5 gram per cubic meter, and the data that we'll review shows even better performance than that. As you can see in this process, we're showing the DoverPac on the left being filled from a dryer discharge, and on the right going back to a reactor for additional chemistry. The beauty of the DoverPac is on the left it's filled and then it's just lifted, transferred to the next area with no inverting, no costly lifts, no split butterfly valves or other valves to aid in the containment, connected and discharged. It's quite an easy system to use. Besides the containment aspect, which is really the critical part, other benefits of FIBC technology include standard sizes, whether it's a bulk handling process or low volume HPAPI. The DoverPac system follows the critical design standards of contain at the source and no open nozzle charging. Again, instead of having isolation technology or local exhaust ventilation technology, we follow the idea of contain at the source so that all of the powder stays within the process, we minimize the exposure to the operator and to the environment. One of the big keys of using flexible IBC technology is the flexible wall and to handle pore flowing powders. This is often overlooked in a critical aspect of using a DoverPac. Here, even very pore flowing powders, whether they're sticky type of powders or maybe very light in density and don't flow well, the operator or some automation can be used to manipulate the sidewall to assure all of the powder is discharged. And there's confirmation of that either manually or again through automation to assure that there's 100% discharge. This is quite difficult on the standard typical technology of stainless steel IBCs, and often containment has to be broken to see if the bin has been totally discharged. Other aspects of the DoverPac, the static dissipative ArmorFlex film, very critical. Sampling capability, we'll take a look at what that is. We can dock a DoverPac to a system without having to be in a complete vertical position. A lot of cases in reactor charging, there could be a drive for the reactor agitator in the way. Using FIBC technology, you can dock at a slight offset and so forth, where IBCs cannot do this. And then there's certain certifications that the DoverPac carries that are critical in many regions and many processes. So from a quality by design standpoint, the DoverPac was developed specifically for pharmaceutical powders. This isn't a derivative of some other product that was brought into this space, but it was developed specifically for high containment of handling pharmaceutical powders, with the key part of it starting with the ArmorFlex film and the quality of that ArmorFlex film, which we'll show through some details in another slide. Standard sizes, again, developed for small batch sizes to bulk processes up to 2,000 liter bulk processes. The static dissipative film and solvent resistance of the film, the ArmorFlex, again, built for powder handling. So it's not a derivative product that's brought into the space with shortcomings. It's really built specifically for this application. And the shape of the FIBC, well thought out at the beginning. The repose geometry, or if you're familiar with the development of hopper designs to allow for good flow, we know that the bottom of hoppers need to be angled at a particular angle, the repose angle, if you will, to get the best flow. The DoverPac system also was developed that way to facilitate the flow, again, assuring that we can get 100% discharge and a nice mass flow transfer from the DoverPac into the next process. Finally, a lot of effort put into the design for safety factors for lifting and loading. You'll see through the manufacturing process, the integrity of the system is taken care of, but when it's being used in a pharmaceutical plant, high levels of safety factors have been applied to assure that there's no failure in use. And then also part of the quality by design was to understand the operator use. The DoverPac does require manual operation from operators, but the idea was to create a system when filling the DoverPac and discharging the DoverPac that the operations were exactly the same, so the operations would be repeated, and this allows for great operator efficiency and enhanced use. Again, the DoverPac system, as we can see, is a closed charging system, very key, contain at the source, again, the design philosophy. So when we say that, the picture on the left shows a DoverPac that has already been discharged into a vessel, and the shower cap, if you will, remains in place until the picture on the right, where the operator will dock a new DoverPac to discharge the next batch and then remove that shower cap through the bag out sleeve. In this process, the nozzle or the charge point is never left open and always protected. Key for containment, but also key for cross-contamination protection. This is really of great value as we look at more and more multi-use facilities, and cross-contamination becomes an extreme key. Also, it was understood that many of the processes the DoverPac would be used in and are used in today require sampling. An example of that would be a micronizer operation where typical micronization would require multiple samples, perhaps at the beginning of the run, end of the run, possibly even on a regular interval every 15 minutes or every 30 minutes. So the DoverPac was designed with an integral sampling sleeve that a sample could be taken at any time, and this would be a contained sample in the sleeve as shown on the right. We'll see later in crimping that there's a seal and separate to allow that sample to be taken away and analyzed at a lab without any exposure at all. So this is part of the whole design process where it was built for a pharmaceutical application, knowing that sampling is always a key to assure the process is in line. And then we have the manufacturing side of it at ILC Dover in our factories in the United States and in Ireland. So it was understood that in general FIBC big bag technology that did exist before 1997, that a lot of the welding processes were such that a high level of failures could occur, and that could not be allowed when dealing with pharmaceutical APIs. So we invented the lap seam welding technique, which changed the weld areas from the weakest point of the assembly to the strongest point of the assembly. The lap seam welding is overlapping 1 inch of the material and welding it together. So in essence, at the weld seam, we have double the thickness in most of the strength. We also have automated the whole process from pattern cutting to piece part matching. The welding equipment is PLC controlled for time and temperature. So the entire process is put together to assure that the product and the integrity of the product is the same time after time after time. And the final test, as shown on the picture in the left, is we do an inflation dwell test to ensure the final assembly integrity, that the welds are strong and there's no defects in the film. Documentation in the pharmaceutical industry is always key, and ILC Dover, with a history in the space industry, was very, very strong at documentation, and we followed through with that for documentation in pharmaceutical. So here you can see an example at the right, our certification of conformance, and this comes with each product to assure the product was built to the specifications, and then the regulatory requirements that it followed are also outlined in the certification of conformance. Through the documentation at ILC Dover, we have a completely traceable quality system to allow for any review that needs to be done, any questions that are brought forward. In fact, this quality system has been audited by dozens of major pharmaceutical companies, and we've never had any major findings, and we've never had any failed audits. So all part of the documentation, from the certification of conformance to packaging of the products in sealed bags with labels, with traceable data, drawings for all the products, manuals and document package to show how the assembly is done, the ArmorFlex material, complete data book on all of the specifications, physical specifications, regulatory specifications. So very heavy on documentation to support the needs of the pharmaceutical industry. And lastly, back to the ArmorFlex, a full range of regulatory standards that are met from a global standpoint, and also then the needed physical properties. When we look at the physical properties, we're really looking at that built for purpose for powder handling in high containment for pharmaceutical processing, including some critical aspects of being able to inert the DoverPac with nitrogen, or to create a low relative humidity level. Very, very important in some applications. So again, the ArmorFlex was designed with only the pharmaceutical market in mind. So now we go to the actual applications of the use of the DoverPac, and we start with the chemical synthesis process with the DoverPac. And again, typical chemical synthesis of an API has the reaction stage, an isolation stage, a drying stage, and then it's often repeated. So as we can see here in demonstration with pictures, early in the stage with the reactors, a DoverPac can be used to charge the reactor and keeping the nozzle closed, which is key. We know many companies have policies that they do not want to have open manway charging at the reactor. We also know from experience with FDA inspectors that they have a preference to have a closed nozzle and no open manway charging. So even starting at the reactor with non-potent materials as the chemical synthesis process starts, the DoverPac is extremely valuable. And then from there into isolation, where a centrifuge would be used, a peeler centrifuge type of system, discharge into the DoverPac, which then can be taken to the dryer, discharged into the dryer, and the dryer discharged out into a DoverPac. And again, with the idea that in any of these steps, we might go back to the reactor for more chemistry. In fact, it's very typical in the chemical synthesis to have multiple steps of going back from centrifuge, from a dryer back to the reactor, having additional chemistry done. And so the DoverPac was set up with that in mind to be able to hold materials, even wet cake from the centrifuge, to hold materials while waiting for the next process. So the chemical synthesis, very straightforward. What we were trying to accomplish is to have a direct transfer between centrifuge, dryer, and reactor, being able to do the chemistry steps while always having a closed manway, while being able to hold materials from dry powders, potent powders, non-potent powders, and wet cake that might come from a centrifuge. We do have some exceptions, so not everyone has the same type of equipment. So in the chemical synthesis process, also often we see basket centrifuge. So on the left, if you have one of these nice basket centrifuge, it's really not possible to use FIBC technology, but it's not over in terms of containment and isolation. And so as you can see on the right, two centrifuges of that type, the basket centrifuge type, are completely contained using flexible isolators. Flexible isolator systems will be covered in a future virtual trade show that we have. But again, just having a basket centrifuge does not eliminate the idea of having high containment. It's just a different technology that we would apply from the DoverPac technology. And then in the same idea, tray dryers, the tray dryer challenge, and for the most part, many processors see that containment of a tray dryer is a very complicated and difficult process. There are some ergonomic challenges, particularly as we get to larger tray dryer systems, but the reality is that many chemical synthesis processes go into the tray dryer in a final stage or in an intermediate stage, and so there needs to be a way for containment. So again, we apply the flexible isolator technology to that, which again, we'll cover in detail in a future trade show. But tray dryers, all types of dryers, very much possible for high containment. It's just when do we apply a DoverPac technology or when do we apply a flexible isolator technology. Moving on to after chemical synthesis, we move into the oral solid dosage processing. And here we're really looking to contain the transfers between each process. And the full range of processes from wet granulation, dry granulation, direct compression, hot melt extrusion, some processes that aren't shown in this diagram, encapsulation, all of these can be contained in the transfers between step to step to step with the DoverPac technology. And we'll show some examples of that for some of these steps. So here we have the example of a CoMill process. In this case, this was a demonstration to prove for a customer application of taking a material in a DoverPac and feeding it into a CoMill, in this case, a U20 CoMill from Quadro. And then after milling, it's discharged back into another DoverPac. The DoverPacs were very unique to be able to do this process without any modification to the CoMill. CoMills typically will have a tri-clamp connection on the inlet and a tri-clamp connection on the discharge. That facilitates the hardware connection for the DoverPac docking extremely well. So here, without modification to the core CoMill, we were able to apply the high containment DoverPac system for this milling step in oral solid dosage. Later in the program, we'll show this picture again and show the SMEPAC study that was done with data for the end result of what the containment was in doing this milling step. Another oral solid dosage process that we've looked at and completed. On the left is a weigh and dispense type of system where material was received in drums and needed to be subdivided or dispensed to a specific weight in a DoverPac so that it could be taken to the next process to be discharged. So here, combining flexible isolator technology with the DoverPac allowed for the subdividing of drums to a DoverPac for a contained transfer in the oral solid dosage process. You know, very key in handling this. In a lot of cases, this process is handling a drug substance. And so now we really have a high containment challenge in dealing with 100% of the active drug versus in downstream processes where the excipients have already been added. The containment challenge in looking for the drug substance or the API is a little bit different. But here, always a critical application in making sure that the transfer to the DoverPac is high containment. The picture on the right is a little bit of a different application where this customer had a policy of no open reactor charging. So we touched base on that before. So here we are transferring drums of material, multiple drums into material to a very large DoverPac, a 700-liter DoverPac. And the idea being, why take 6 drums to a vessel to discharge all 6 drums, which is very time-consuming, along with having an open vessel. Why not transfer those into one DoverPac and make one discharge into that vessel? So very efficient when it comes to using a DoverPac for dispensing. So our containment assessment number 1 is, again, the project here was that ILC Dover worked with a third-party industrial hygiene company. The DoverPac system was used. Again, when we talk about a system, it uses our docking hardware, the O-ring canister. We have special clamps that are used. There's a technique called the clean zone, which further assists in assuring the best possible containment. And then the seal and separate method, crimp lock, which we'll touch base on as we go through this. It's very key to use the proper seal and separate method for containment at the point of separation, but also further risk assessment. This is a 700-liter DoverPac, and the surrogate was lactose. So here's the data, one of the data tables. Now this was from discharging the DoverPac, so charging into a vessel. And as you can see in the typical SMEPAC protocol, there's 3 iterations: run 1, run 2, run 3. And in this case, the data table summarized the geometric mean in micrograms per cubic meter. So in this case, the thing to note closely is on run number 2, on the second personal breathing zone, that we had a reading of 0.23 micrograms. And this is the highest reading we'll see in this SMEPAC test. And then we'll take that data and we'll take a look at what does that mean with the actual containment performance of the DoverPac system. We also, in this test, we looked at discharging the vessel. So after the first test where we filled a vessel, then we discharged that vessel into a 700-liter DoverPac. And again, the data table, same concept of 3 iterations of the run. And the data is quite good here with obviously anytime we see the less than number, that's referring to the level of detection. We were below the level of detection when the air sampling was done. So our readings were extremely good, and seeing the arithmetic mean or the geometric mean, the numbers are extremely low for the level of containment. And lastly, we looked at the seal and separate process. So here was sampling, air sampling around the seal and separate process. Traditionally, before ILC Dover brought the DoverPac system forward, the standard was a twist, tie, tape, and cut for separating an FIBC or a big bag product. And that's very operator sensitive. The twist and the tie can open up over time and so forth. So it's very key. As you can see here, again, we went through 3 iterations of the seal and separate, and we have the geometric mean again, which is very nice, very good, very low numbers. But this is very key to examine the seal and separate. This could be a worst case scenario in a lot of cases for operator exposure when a seal and separate system is not as secure as the crimp lock process. And the key to that is if there is any residual gas left in an FIBC, that can cause for an aerosolization of powder through a poor seal. Let's say a seal that wasn't as tight as a crimp lock system allows for. And that aerosolization can cause operator exposure. So it's very key to look at the entire process through the system. So in this study, we looked at filling a vessel, discharging a vessel, and then we looked at the seal and separate process so that we were sure to capture any risk along the way of operator exposure. To sort of reverse analyze this from a mathematics standpoint, we looked at the data, and as I had noted, our one reading was a 0.23 micrograms per cubic meter. So that was our highest reading. So we would take that and say, okay, we need to do some analysis around that. But taking all of the data in the test, we would do a geometric mean statistical analysis that would give us a 95% confidence factor. And that statistical analysis would show us 0.13 micrograms per cubic meter. So different companies, the pharmaceutical companies have different rules. There are some industry rules that are followed in some cases. So one of the industry standards is pharma companies will use a 50% rule. So in using the 50% rule, we would look at the 0.13 microgram of the geometric mean and double that. So we're reversing the application. Typically, the OEL is set, and then you go down from that. But we're doing this in reverse based on the data. And so this would say that our containment performance target of the DoverPac would be 0.26 micrograms. In a more strict application of that, applying the statistical analysis of EN 689 in the 2018 version, the new version, which speaks to the containment performance target has to be at 10% of the OEL, if you will. So here, that would take the containment performance target to 1.3 micrograms. So we would our 0.13 is 10% of that. So applying that strict rule, we get a bit of a different look at, well, what is the containment performance target? Now, essentially, different companies have different rules to look at, well, what is the OEL, and then what is the target containment, and so forth. So having an understanding of how the rule is going to be applied and mathematically how to look at the containment is very critical. And again, actual containment performance is based on characteristics of the powder, the process, and the volume being transferred, the mass transfer of the powder. So there can be some variance, and also, as we said, the process. As we looked at milling is always very difficult from a containment standpoint. So here's that CoMill again. So we took this system and we did the same kind of HPAPI study on this. Using, again, a third-party industrial hygiene company, using the DoverPac system. Here, instead of using a 700-liter DoverPac, we used the 110-liter DoverPac, and in each time, it was filled with 25 kilograms of the surrogate, and the surrogate was lactose again. And the data here is very similar to what we had on the 700-liter system. We can see that in one of the breathing zones, we had one reading that was at 0.35 micrograms per cubic meter. But when we look at the geometric mean analysis of it, we're getting, really good numbers again, well under the 0.5 micrograms per cubic meter that we started the presentation with. What this shows, the test data shows consistent results, and ILC Dover has performed dozens and dozens of HPAPI studies, and one of the things that we're confident in is the data is always very, very consistent. There are outliers in some of the tests that have been done, and so we look to mitigate that through operator experience, training, and so forth, and to continue to evaluate the system to see any gaps where improvements can be made. And as noted, milling systems are a containment challenge because milling tends to add a lot of energy and fluidization of the powder, which can make the containment more challenging. CoMills present a small challenge. Hammer mills can present a much higher challenge. So again, understanding the process, understanding the volume of powder, understanding the characteristics of the powder are very key when establishing what the containment performance of these systems can be. So moving on a bit, we wanted to compare the DoverPac, or in general, single-use products, to rigid stainless steel products. So we're going to look at a brief analysis as we go forward in the DoverPac versus a stainless steel IBC product. But before we do that, we thought we'd borrow some information from the biopharma industry, where there is a massive reliance on single-use products, and this is as much for the process than containment. But the biopharmaceutical industry has a tremendous amount of information that's important when we look at single-use products and comparing that to stainless steel products. So there's been a lot of life cycle analysis done to show the significant savings. And here we're looking in this table that we're referencing from a Thermo Fisher paper that had a significant utility savings, and we're looking both at water consumption and energy consumption. And this is really through a lot of the cleaning processes and so forth. So if we look at a full life cycle analysis, it's very interesting on the level of savings that can be won by going to a single-use product. Cleaning validation, hold times, all slow the production cadence in a manufacturing operation. And all of that is needed when you're using a stainless steel type of product. Single-use products eliminate a lot of that and minimize the risk of cross-contamination from retention. So the biopharm industry looks at it from a different aspect, but in the pharmaceutical manufacturing, we look at retention as described in the ISP risk map document, which is really the number one reason for cross-contamination is retention on a surface after cleaning. And so going on to the next batch, that retention gets into the next batch and potentially is that cross-contamination. As we look at the DoverPac versus a stainless steel IBC, we actually remove 100% of the risk of retention because the DoverPac is a completely disposable single-use product. Before jumping to that analysis, we don't want to forget about drums. There's drums everywhere out there, and drums require significant manual labor. And what we see in most of the applications where they're being used is there's an overall lack of containment or containment through isolation or ventilation systems, localized exhaust ventilation systems, and that type of thing are used. So again, as we had referenced earlier, anytime we can get out of using drums and getting the drums in a contained way into the DoverPac, process efficiency is going to be improved and so forth. And we see all of these different processes going on from, on the left, again, a sampling operation in a drum where the DoverPac has a sampling sleeve. The middle picture, we've got an inverter that's inverting into a bin with drums, very messy. Often there's a lot of powder that escapes into the environment as this process is being done. And lastly, the gang filling of drums, just not really a lot of containment there. And again, in this picture on the right, the operators are relying on PPE for their primary protection. And as we always note that in the regulatory in regulatory writings such as OSHA in the United States, that PPE should not be used as a primary containment method. And through the hierarchy of controls that we all know, that an engineering control in a DoverPac would comply with the idea of an engineering control. The engineering control is preferred whenever it's possible. So again, back to that comparison of IBCs versus FIBCs, and really we just wanted to run through, what does that mean in terms of operation costs and so forth, and comparing the cleaning versus disposal. So the typical process on a stainless steel IBC starts off with there's an unknown amount of powder inside the IBC because we can't see it, or we have to break containment to see it. So we don't want to break containment. So there is some unknown amount of retained powder, which is inactive. And so that rinse, that first rinse, is always captured for disposal. And that when we say capture for disposal, typically that means it's going to incineration. So again, not very efficient to have to burn water, if you will. And then on to wash with detergent and another rinse. So that's either treated or sent for disposal. Again, disposal would typically be incineration and so forth. Then we have a drying period, which has additional energy costs for the desiccated drying of the inside of the IBC. Typically, again, if we're working with high containment systems, there has to be a containment valve of some sort, like a split butterfly valve. That will always have to be removed for complete cleaning of the seals and maintenance of the seals and so forth. And then there's a lot of other hidden costs with the IBCs in this cleaning process, including that space needed for either a bin washer or CIP stations. And this is dedicated floor space. So we know in a pharmaceutical facility, floor space is at a premium. And if we could eliminate the use of cleaning a large bin washer or a large CIP system, it frees up a lot of floor space. And then we have just the floor space for storage of the IBCs when they're not being used. So lots goes into the idea of cleaning the IBCs. This needs a lot of attention from labor, materials, and then floor space for storage. On the FIBC side, this is very sort of simplistic. After the IBC is discharged, typically there's a process to evacuate gas from the inside. There might be some retained gas, so we don't really want to create a balloon, if you will. So evacuate the gas through a simple HEPA filter, seal and separate using the crimp lock system, and now it usually will go into a drum for disposal. So DoverPacs typically from a pharmaceutical process are incinerated. So the polyethylene-based film, along with the polypropylene-based outer restraint for support, has a minimal carbon footprint, and an actual analysis, mostly again coming from single-use technology in the biopharm market, the overall carbon footprint is significantly less when using single-use products versus the overall carbon footprint of developing, manufacturing, transporting, and cleaning products made from stainless steel. So a lot of evaluation has gone on in that area that really needs to come over to the pharmaceutical industry to see that operational costs of using single-use technology is very much desired, and when under analysis, it proves the value of the single-use technology. So here we'll look at the capital cost in a typical IBC versus an FIBC. And so we just put together a scenario. You can see the notes at the bottom. Again, assume a product contact of 316 stainless steel for the IBC technology. We're looking at a containment performance target of less than 1 microgram per cubic meter. We're saying that because there needs to be a containment valve when we're dealing with less than 1 microgram. Perhaps if we're dealing with something above 10 micrograms, a standard butterfly valve could be used, which could save money, but there's not really containment. And then the calculation for this is 6 batches required 5 IBCs or 5 FIBCs per batch. So those are the numbers we use to come up with this brief analysis. So you see the stainless steel IBC on the top. We've got to purchase the IBCs, the high containment split butterfly valve. How many are needed? Really good question, because at the analysis we said 6 batches requiring 5 IBCs or FIBCs. You're always going to have IBCs in hold time for wash, what have you. So if the batch size needs 5, oftentimes the purchase has to be closer to 10 or more, so that there are excess bins to be in the process of cleaning, hold time, and so forth. And then we have to go into a purchase of precision post lift, because using an IBC with a split butterfly valve, we need to dock in an extremely precision way. And here we estimated $125,000 for such a post lift. Oftentimes they're even more sophisticated than that, and so that cost goes up. washing machine, which could be a bin washer, CIP system, and so forth. So all of those come out to, in this analysis, about $650,000 to be ready to run this process of 6 batches requiring 5 units. Whereas on the DoverPac and the bottom analysis is purchase the high containment docking system. So typically 1 for filling and 1 for discharging. So this is the CapEx of $40,000 for 2 of these high containment docking systems, which are our O-ring systems. And then a simple hoist to lift. And again, that's one of the keys, that a DoverPac can be lifted with a simple hoist system. We don't need a sophisticated post-lift kind of system. So really closer to $50,000 to buy a stainless steel grade simple hoist system. And then lastly, the high containment DoverPacs, and roughly $600 a unit for the high containment DoverPac. And you buy these as you go. And the beautiful thing is they can sit in the stores. There's a 5-year shelf life to this, so they don't take up much space. And they're available to be purchased just as they're needed. So we looked at this analysis, and we came out with a CapEx cost of $108,000. So a dramatic savings on the CapEx cost. And again, as we referred to when considering the complete use of an IBC versus an FIBC, the OpEx cost does not ever catch up to the CapEx cost, because in the operational expense, there continues to be a benefit in using a single-use technology. And then we look at project time and complexity. So starting on the right, we've got the IBC project. So in a project, the complexity of it, we have to select how many IBCs, the size, we have to select the containment valve, the precision lift, we have to decide how the cleaning process is going to go on. We've got to manage that floor space, because we're going to have a lot of dedicated floor space to post lifts, to storage of bins and things like that. We've got to manage controls, including the electrical classifications, because when we're using this type of equipment with a post lift and so forth, the electrical classification of a room is going to be very critical. Again, we'll see that we don't need that with an FIBC project. We have to manage the FAT and the SAT, operator training, validation documents, and more. And so roughly a typical project using a stainless steel IBC could be up to 30 weeks. Where on the FIBC project, we select the FIBC size, okay, and units can be purchased as needed. So they're available to be purchased as needed. It's not a hard asset that you purchase. It's a single-use product that comes in as you need it. No containment valve, the DoverPac system contains itself. That simple hoist that we were talking about, which means there's no floor space required for that. An FAT and an SAT typically take 1 day, because the system is so simplistic. Operator training, 1 day, and the validation documents somewhat minimal. So we look at about 10 weeks to complete an FIBC project from start to finish. But there's also the other considerations, and recognizing the change in the pharmaceutical world from 10 years ago. Maybe we should go back further. 20 years ago, there was master site planning, that a pharmaceutical site the overall plan would be an extended plan on what products the site would make and what the size of the batch sizes would be. We really don't have master site planning anymore. It's really over the next 3 to 5 years. What are the projections? The sales projections could be for very high sales of a drug compound. They could be for maybe lower, which either way could impact the overall use of the equipment and so forth, which then leads to tech transfer. So if a drug product has a very good value on the market, additional manufacturing through CMOs or transfer to international operations could be needed. So again, very easy to do a tech transfer with the FIBC, the DoverPac system. The IBC, again, we're back into fixed assets that require another project, another management to be to go through, and another CapEx expense. So we really look at the project time and complexity and those things that affect that over the long term. And again, an FIBC project is very quick to implement and can be transferred as needed globally. So that was the content of our webinar today. So I thank you very much for attending. Again, as we said, we'll have time for your questions, but before going, watch for our next webinar planned for May 6th, where we will discuss flexible isolator technology throughout the pharmaceutical value chain. Again, there will be some applications in chemical synthesis and then a lot of applications in oral solid dosage. So with that, I'll throw it back to our host, Paolo, and Paolo will address some of the questions that have come in.
Yes, thanks, Scott, for speaking about this technology and how this flexible containment and the DoverPac can help on the manufacturing process. Basically, and I'm just trying to catch a few points. Basically, the DoverPac can accelerate the transfer of powders into the process. And if we look to the drum charging inside into a reactor, it's a quite different way, because we will have an FIBC and a full discharge. It will accelerate our production. It will protect somehow our patients, because we are reducing the risk of cross-contamination, protecting our staff, which basically means bring ourselves and our colleagues safe home at the end of the day. And last but not least, it prepares the facilities, especially when we are talking about shared facilities, for the future challenge, make easier to accept new products, providing manufacturing flexibility, having less time equipment stopped for non-predictive operations like cleaning or validation. So, and the first question, I will try to do 2 questions in only one, which is really, I would say, are really complementary, and talk a little bit about the documentation. So basically is asking, during the introduction and presentation, you talk about the material being compatible with a range of solvents and meet all international standards. Where can we get this information? And the second part basically is asking, how do you resolve the electrostatic problem with an API that can be adhesive to the FIBC and cause some problems with uniformity of content? Have you faced such a situation?
Yes, I think from both parts of the question, I think we've experienced this from several processors. In terms of the solvent resistance, again, the film, the ArmorFlex film, was developed with that in mind, and so we selected polyethylene materials that are very resistant to the typical solvents that are used in pharmaceutical processing. And so that information is contained within the data book, which is available to our customers. In the data book, we've gone through and done testing for more than 24 typical solvents used in pharmaceutical processing. We quantified the testing. Well, first of all, we used a standard ASTM test to do the testing, and then we quantified the testing by doing the submersion of the ArmorFlex material in a 100% solution for 48 hours. So the data is very compelling, because we did quantify it, and most of the solvents had less than a 10% impact on the physical characteristics of the ArmorFlex. But all of that is summarized in 2 nice data tables in the ArmorFlex data book, which we would urge our customers to reference and look at to know the methodology that we use to create the ArmorFlex and to make sure we're addressing the solvent issue. On the second part, the electrostatic charge and the surface adhesion, if I understand the question correctly, it's really surface adhesion of the powder to the inside of the DoverPac, and as the discharge is being done, that surface adhesion could hold residual material into the DoverPac. And this, again, is the beauty of a flexible wall technology. We always suggest at the end of a discharge that the operator will physically take the wall of the DoverPac and shake it, just shake it like they're shaking a blanket, if you will. And we found that this generally releases almost all of the surface adhesion through an electrostatic charge from the wall of the DoverPac. Again, the DoverPac has a static dissipative effect to it. So by nature, it does not want to build up a static charge and hold particles, but there will be some surface adhesion. And so by shaking, it will release that.
Yeah, thank you for the explanation, Scott. On the next, we will talk a little bit about robustness and some properties of the connection. So basically, someone is asking if what kind of automation do we need to ensure that the DoverPac is full discharge when we are working with poor flowing powder, and when what happens if not how to ensure that it's not damaged, isolate, and how can we ensure that we are not damaging with the kind of automation the DoverPac discharging.
Right. So the automation in charging is typically straightforward, even with poor flowing powder, that the DoverPac can be placed on a scale. So using a known weight, then the DoverPac can be filled. The operator can monitor that as the filling is going on, knowing that, let's say, the target weight is 200 kilos. As approaching 150, 175 kilos, we suggest that the flow rate in the mass flow rate of the powder into the DoverPac is restricted a little bit. We don't want to overfill it, and that is a key, is not to overfill the DoverPac. So the number one way our customers will control that is placing the DoverPac on a scale and/or having a dosing system that will control that flow into the DoverPac and automatically shut off when we reach a target weight. So I think that's key in terms of discharging and looking at automation to make sure that it's a 100% discharged, and also from an integrity standpoint of the DoverPac assembly. So the automated way to do it is really a bag massager, which are quite common, used in a lot of processes outside of the pharmaceutical industry. And using a bag massager, particularly in those markets, let's say the food industry or the plastics industry, those systems are not nearly as robust. The big bags are not nearly as robust as a DoverPac. So we've really designed into the DoverPac from the outer restraint to the strength of the ArmorFlex film, that even when using automation to assist in powder flow, that the integrity of the system is going to still be very safe. So I think, again, that goes to the quality by design. It's not just the ArmorFlex film, it's the lap seam welding, it's all of that goes into the quality by design to assure that there's not a problem when using automated discharging.
Thank you. Now we are moving to the environment question, and from a sustainability point of view, and taking in consideration that we have now facing a global challenge to reduce or eliminate use of plastics, how can single-use technology compare to stainless steel IBCs or even rigid isolators? And I will add a second question on that. What is the disposable policy for typical DoverPac?
It's easier to answer the first question or the second question first, and then move back to the first question. So the typical disposal of a DoverPac in the pharmaceutical industry is incineration. We see the majority of our customers will take the DoverPac after it's been used and place it into an empty drum, and that drum would go to whatever facility they use for incineration. Secondarily, we do know that some customers would go to landfill with that and probably treat it as a hazardous material, because again, we're typically dealing with potent products and so forth. But incineration is the #1 methodology. Again, the material being used in the DoverPac are polyethylene based. And so the carbon footprint that we're going to leave is generally fairly low. And then when we take a look at some local regulations and so forth, we don't have to deal with some of the issues that other materials and maybe in flexible isolator technology deal with in using materials like polyvinyl chlorides and things like that. So we're not releasing chloride gas or anything during incineration. So I think incineration is considered to be the most optimal way to go to remove the hazard of the compound, the residual compound that might be in it, and not have to use a landfill. The first part of the question is really a part of a complete analysis that must be done, and it goes to the first idea. The simplest idea was from the example we borrowed from the biopharmaceutical industry. Is when you're dealing with stainless steel systems and you're working with a lot of CIP and a lot of cleaning, there's a tremendous amount of water that's being used, and there's a tremendous amount of energy that's needed for that water. And one, converting it from water to WFI, so that's a significant cost to do that, and the energy costs associated with that. So the analysis that are becoming more and more prevalent and really born out of the biopharmaceutical industry take into consideration all of those things, from converting water to WFI to use for CIP, doing the CIP, then incinerating that water. It's not very green, if you will. And so environmentally speaking, even though the idea of using a petroleum-based product, a plastic, if you will, single use and incinerating seems to think as though that would be the less ecological friendly process, all of the studies have shown the exact opposite, that all of the energy and all of the costs associated with cleaning makes the single-use products more desirable from a cost standpoint and from an environmental standpoint.
Yeah, thanks, Scott. So to be very honest, we have a very high volume of questions today. And due to the time, we won't be able to answer to all, unfortunately, but we will provide you some answers by email very soon. I would like to emphasize that containment is a very complex matter, as you understand, and data is base for getting successful results. So just to have an idea, when we talk about 10 micrograms of API, it represents around a sugar crystal divided by 20. So the human eye will never see that. So the limit of the naked eye is around 5 to 50 microns. It means that when we talk about containment, it's necessary to be very scientific, working with high reliable data, complete risk assessments, and especially avoid the common sense such as, well, you know, my process is really contained. You won't see powder anywhere. So today, the challenge is there is no detector able to get a feedback in real time, contrasting with other dangerous materials that we are facing every day in our plants, which makes handling potent powders a highly risky job with severe consequences if something fails. This makes containment a huge and a big priorities challenge to all of us. And the use of a reliable technology associated to a strong training program makes the total difference and creates confidence and trust from operators to senior managers to shareholders and customers. I would like, before ending, thank you very much for joining this webinar. It was a big pleasure for Scott and I to share with all of you this presentation. And as Scott said, hope we can see you soon next May 6th, where we will talk about flexible isolator technology. And if you have any further questions, comments, or you want to share some experience about our technology or other technologies, please don't hesitate to contact us through our regional sales manager or by using our webpage, www.ilcdover.com. Please keep you and yours safe. Thanks again and have a great end of day. Goodbye.